All right, thank you very much, and I appreciate this invitation and this opportunity to tell you what we're learning about the conformational changes at the heart of RNAi. And the work I'm going to describe is work biochemistry done by Peter, a student in my lab, and then the cryo-EM by Abdallah, a student in Seychelles' lab. And as a disclosure, as Mano mentioned, I have a financial interest in Alnylam, and who also is quite interested in RNAi. So our lab is interested in post-transcriptional gene regulation. Why is it that some mRNAs are 1,000 times more stable than others? Why are some translated so much better than others? And part of this has to do with these microRNAs. And these are small RNAs that are processed from hairpins. They associate with an Argonaute protein, and from there they downregulate many mRNAs. Most of the human mRNAs are conserved targets of these microRNAs. And this targeting is primarily through pairing to what we call the seed of the microRNA, nucleotides 2 through 7. And when this happens, the Argonaute will recruit indirectly these deadenylases, which shorten the poly-A tail. And then in most contexts, except for oocytes and early embryos, the shortening of the poly-A tail causes decapping and decay of the mRNA. Now, there's also this other pathway that we all know about, where if you have very extensive complementarity, not just the seed pairing, but very extensive complementarity to the target, and if you have the right Argonaute, in humans Argonaute 2, then you get slicing. And this is not very common in our cells. We only know of 21 different endogenous transcripts that are sliced by this pathway. And there's a few human viruses that also slice transcripts. But, of course, this turns out to be a very convenient part of the pathway because when you design siRNAs, as Tom Tuchel first did, to feed into this pathway, then you can have this gene knockdown tool, which of course has been had a huge impact on biomedical research and is being used now to make therapeutic compounds. So today I'm actually, because of the nature of this meeting, I'm going to focus on this side here. Our lab has done a lot of work on the microRNAs and this here, and of course the siRNAs also can do this. We call those the off targets. But what we really want to talk about today, it will be the on-target activity, the slicing activity of the siRNAs or the microRNAs or the guide RNA. Basically, those are going to be interchangeable for this talk: siRNA, guide RNA, microRNA, because once they get associated with AGO, it doesn't really matter. It uses them all the same. All right, so one question that we've had is how do you get to this fully paired conformation? So as you know, it's not that railroad track that I drew before. Instead, it's a helix. Okay, and so we're starting with the Argonaute and the guide RNA. And in the slicing activity, when we're talking about the slicing activity, this is often called the RNA-induced silencing complex, or the RISC. And so we have the RISC, we have the free target, and then the question is, how do you get to this 2 turns of a helix, fully paired, catalytically active conformation? And initially, we all thought it was pretty simple. You just have pairing to the seed region here of the guide RNA, and then pairing can just propagate to the end through sort of this zippering type, nucleation and propagation, very simple. And we were quite fond of this model. In fact, the reason that we called this region here the seed is because it's where the pairing starts, and we thought that for more extensive sites that this would be where the pairing nucleates. However, in thinking about this more, we realized that this is a bit challenging. Okay, the seed region here, this helix is stationary, it's immobile, it can't move or turn or anything like that. And so if you're going to get pairing throughout the remainder of the guide, then it has to happen on one side of the seed here. And this guide RNA is attached to the protein not covalently, but it's bound here at the pos domain, and also you have contacts throughout its length. And so it's going to be hard for this target RNA to somehow wrap itself around that guide RNA. And so we thought that, well, perhaps what's happening here is instead of just propagating the pairing from the seed to the end of the guide, maybe what's happening here is that you have the target pairing initially to the seed, and then you skip over the central region and then pair here to the supplementary region. And now you have that second helix. And that second helix is not immobile or fixed. It can move. It can rotate. And as it rotates, it can just sort of rotate over the pos domain, over the end domain into the central channel here, and as it rotates, you get the twist needed to pair into the middle of the guide here, into the central region. So when the one helix rotates, you can get pairing propagating through the central region, and then pairing eventually, or either before or after pairing at the ends. As it rotates, you sort of lose these contacts between the guide and the protein. All right, so this is the way that we imagine it now, and just to have a picture in your mind on how we think this is happening, I just want to show you this morph, okay? And this starts from the two-helix structure, okay? And these are crystal structures from Ian McRae's lab. The first has a shorter second helix and then a longer second helix, and then it morphs to the structure I'll be talking about later, which has the fully paired catalytically competent duplex. Okay, so we're going to do the morph here, and the RNA, again, is using the same colors. So the guide RNA is in red, the target is in blue, and you can see these two helices here. This is the seed helix, and then here's the second supplementary helix. And this one is not going to move, and this one will be the one that rotates. And you can see how the target is just laying on top of the guide, okay? Nothing tricky, nothing difficult here. It doesn't have to wrap itself between the guide and the protein. It just lays on top of this region, and then comes in again and lays on top of this region. Okay, so that's very easily accessible, and we see this for about 5% of the microRNA targets, this seed plus supplementary type pairing. So that's not going to be a challenge. And so now what we'll do is we'll morph from that two helix, and as we do that, again, this part will be stationary. This will rotate. This part will come away from the pos domain as it. So you can see it rotating here, getting to the fully paired structure. Okay, so that's, again, we can't say that any of the details there are correct. It's just a computational morph, between these three structures. All right, so when Peter was interested in this process and was thinking that perhaps different sequences are going to behave differently in this sort of conformational change in catalysis. And so he decided to measure the slicing rates, the elemental rate constant here for K, what we call K slice, which includes going from this two helix conformation to the fully paired conformation, and then the chemical step of cleavage. Okay, and he does these reactions in large excess of the RISC complex, and so he doesn't need to worry about multiple turnover or product release or anything like that. And then because this binding here to the seed and supplementary region is very stable, you don't really have an off rate, or you don't have to worry about the dissociation of the complex. And so really, the reaction that you see is a function of the association rate and the K-slice. And we can tease those apart by using different concentrations of the RISC complex. So always in excess over the target, but different concentrations, and then we can fit to get the association rate and the K-slice elemental rate constants. Okay, and so previously this had been done by Phil Zamor's lab for 2 different guide RNAs. And what they found was that the slicing rate here was, the K-slice was somewhere around 3 to 5 per minute. And Peter did this with another 13 guide RNAs. And what he found is actually quite diverse, the slicing rates that you get for these different sequences. All the sequences perfectly paired to the target. And so the time of slicing could go as fast as 6 seconds or as slow as 28 minutes. Okay, so we're talking about a 250-fold difference, variability, just depending upon the sequence of the guide RNA. And Peter then was able to find three places in the guide RNA where the sequence seemed to be imparting these differences that were the determinants of this slicing rate. So these positions were position 7, where you had a preference for weak pair. A weak pair makes the slicing faster. Position 10, where a purine pyrimidine, purine here on the guide, enhances the slicing rate. And 17, again, a weak pair, an AU-UA enhancing slicing. And so Peter could then confirm this by mutagenesis. So, for instance, you could take miR-124, which starts off with a pyrimidine here at position 10, change that to a purine, A, and then put in the U in the target, and he would see about an eightfold increase in the K-slice value. Conversely, if you take miR-7, which starts off with a purine, and switch it to a pyrimidine and give it the perfect complement, you get about a 9-fold decrease. Okay, and so these types of experiments were able to support each of these 3 determinants. Now, so then what he did is he made fast and slow versions of 2 different microRNAs, where he would just, by changing these 3 nucleotides, he would make it either faster or slow, and you have the fast and slow versions of 2 different guide RNAs, and then he would look at how well they work for RNAi in cells. Okay, and so in this experiment, the slow version is orange, the fast version is green, and he did this for 2 different pairs. One was based on miR-7, the other based on miR-196, and he did it in different cells, and so it didn't matter, which pair he used, which cells he used, which context he used. He always found that the faster one of the pair was more effective in cells. Okay, so what this tells us is that slicing can be rate limiting for RNAi in cells, the slicing step. We knew that the product dissociation rate could be rate limiting, and that the association rate, the K-on, can also be rate limiting, but also slicing can be rate limiting. So depending upon the sequence, any one of 3 different steps can be rate limiting in cells for RNAi. Well, these results here that he got for RNAi dovetailed nicely with work that was done over 20 years ago by Dharmakhan. And what they showed was that they just looked at a bunch of different siRNAs and their activity in RNAi in cells, and were able to identify, regions and nucleotides of the RNA, positions of the RNA that seem to be important for that slicing activity. And some of this we've known for a long time, like this weak pairing here at the 5' end of the guide. This is important for loading of the guide RNA association with Argonaut. Okay, but there are 3 positions here that were still mysterious as to what they might be doing. And those correspond really nicely with Peter's 3 determinants. Okay, so this non-CG here is a version of a weak pair here. And this AU corresponds to the purine pyrimidine, and this UA, again, a weak pair here. So this tells us that the reason that these determinants here correlated with better RNAi is because of they're increasing the slicing rate. Okay, it's not like the stability or the loading of the RNA or its stability or getting it into the cells. It seems to be at the slicing step that explains these determinants here. So what then would be a reason that a weak pair here at 7 would somehow enhance the slicing rate? Well, what had been known is that in this area between 6 and 7, there's a kink, a distortion in the backbone of the duplex. And if you look in different structures from the Cray lab here, those with more and more pairing, 3' pairing, have more and more of this kink where you have instead of a 3' endo, 2' endo, and different configuration here in this area. And this again correlates with additional pairing to the 3' region. And Alnylam did a nice experiment using glycal RNA, where instead of a ribose, they have the glycal linkage here, which is very flexible. And they marched that across the guide RNA and also across the passenger strand. And what they found is that at every position, having that flexibility reduced the amount of RNAi, except at position 6, 7, where the flexibility actually enhanced the RNAi. Okay, and we also found that mismatches here or here would enhance the RNAi. They really inhibit the initial binding, but once you deal with bound target, those mismatches actually enhance the slicing rate. So that's what we think is happening at position 7. At position 17, even though a weak pair is preferred, when we put mismatches here, we got like an 8 to 64 decrease in slicing. Okay, so this position is very sensitive to the slicing in the context that we looked at. And that was actually a bit of a surprise because there had been this idea that pairing beyond position 16 doesn't really matter, or it can even inhibit the slicing. And so that came partially from this very influential structure from Dinshal Patel and Tom Tuchel, where they solved the crystal structure of a bacterial ago, Thermus thermophilus, with a guide. It actually uses DNA as a guide, and a 21 nucleotide guide RNA, a 19 nucleotide guide DNA, a 19 nucleotide target RNA. And when they solved the structure, what they found was that the pairing only went to 16. After that, it was disrupted by the end domain. The end domain was acting as a wedge, and you couldn't even see these nucleotides in the crystal structure because they were disordered. So that has led to this idea that to get to this slicing centrally paired configuration, you need to disrupt the pairing here beyond 16. So later, when the McRae lab was solving the structure of a guide RNA and a target RNA that again were perfectly paired, they only went to 16 with the idea that you wanted to disrupt the pairs here anyway. And what they ended up with, surprisingly, was not the fully paired, but the two-helix structure, which was awesome for looking at the two-helix structure, but just not exactly what they're going after. Later, they were able to solve, though, the centrally paired from plant, okay, the Arabidopsis ago-10. This is actually by cryo-EM, and they got a mixture, as it turns out, of these two. But by cryo-EM, they could distinguish the two and get both structures. But again, what was happening beyond 16 was unclear because there wasn't anything beyond 16. So these structural studies were all, I think, heavily influenced by this idea that the end domain acts as a wedge to disrupt this pairing. And there's biochemistry that was also suggesting, in some cases, that either the pairing didn't matter at this end or could actually be inhibitory. So Peter decided to look at this. It was kind of murky, the biochemistry. And so what he did is he looked at the K-slice values, either with paired 3' region or with mismatches from 17 on. And what he found was that for some sequences, there is no difference. You get pretty much the same slicing rate. And those turn out to be sequences that have strong pairing here in the middle. However, for sequences that have weak pairing in the middle, so instead of G's and C's here pairing, you have A's and U's, then there was a very strong requirement for the 3' pairing. You would get like if you have all AU here, this can be 600 times slower, or this is 600 times slower than when it's paired here at the end. So a very striking difference between the behavior of the guide RNAs that are pairing with GC-rich pairing here versus AU-rich pairing here. So, and Peter went on to then use chemical probing to look at which conformation these were in. And what he found was that when you had pairing all the way to the end, the chemical probing gave this structure. Okay, it was able to get from the two-helix structure to the fully paired without a problem. And this was highly populated. Or if the pairing's very strong here in the middle, you also had a highly populated centrally paired conformation. However, if you had weak pairing in the middle and terminal mismatches, then the structure was stuck in this two-helix conformation. And that explains why it was 600 times slower. It just wasn't able to get into the fully paired configuration. So this has, I think, 4 important implications. First of all, and if you have moderate pairing in the middle, you also have some requirement for that 3' pairing. So first of all, this tells us why the biological sites, the sites that, I told you about the 21 sites in human transcripts, but also in other animals, there's a lot of slicing going on. And those biological sites, they pair to the ends. Okay, and that's because most of them are not just all GC pairs here in the middle. Okay. It also helps us for siRNA design if you're concerned about slicing off targets. Okay, you're still going to have the issues with seed off targets, but if you're concerned about slicing off targets, then you want to design siRNAs that have weak pairing here in the middle, and that'll enforce the need for pairing to the end. this also, the third important implication is that this coupling between the strength of pairing in the middle and the presence of pairing here at the 3' end, we think is really nice evidence for this 2-helix intermediate. Okay, so the idea is that in this last step where you're forming this duplex, starting with a 2-helix intermediate and having the rotating of this second helix, that all these things that are happening are each reinforcing each other. Okay, so they're promoted by the other steps, the other things, and also promoting. So the idea is that pairing here from the second helix to the 3' end of the guide, that is going to promote, that's going to untether the second helix and allow it to roll. Okay, so it's going to promote it, but at the same time, if that helix is rolling, it's going to dislodge the RNA and promote pairing to the 3' end. And then the same thing in the central region here. As you have pairing into the central region, that is going to promote rolling of the duplex, of that second duplex. And as you roll that second duplex, it's going to promote pairing to the middle. And so all of these things are affecting and reinforcing each other in this model. And that's what we're seeing by this coupling between the pairing here at the end and the strength of pairing in the middle. Okay. And then the last implication of these experiments is that, as we showed with chemical probing, actually this conformation here, this fully paired conformation, as long as you give the RNA base pairing to the very end, then you can populate this conformation and get the structure. Okay, previously it was thought that this was intrinsically unstable due to a need to disrupt the pairs at the 3' end, but that didn't seem to be the case. We can see a lot of sequences, basically every sequence that we tried that was fully complementary was getting to this stage by chemical probing. So we could go after the structure, and so that's what Abdallah did, the student in CSHL Voss's lab. And so then the question becomes, how does the human risk accommodate the fully paired target? So this is our chance to look at that. And in the meantime, we were also encouraged by subsequent work that had come out after the TTAGO in bacteria. So here's another bacterial structure that's fully paired. And it has a guide RNA and a target DNA. It doesn't slice, it just binds to single-strand DNA and inhibits polymerase. And this one they're able to solve it, fully paired, and the way that it's able to get past the end domain here is by a pretty strong bend between 11 and 12. Another bacterial ago, this one has a guide RNA and it slices the DNA. This one, the duplex is pretty uniform, and what happens is that the end domain moves out of the way. Okay, so what's happening with human? Is it this with a bend, or is it this with the end domain moving out of the way? Or is it something completely different, like cascade, okay, where you have all this perfect complementarity, but look at the target here. It never wraps around the guide. Just lays on top, pairing 5 pairs, and then it's disrupted, and then another 5 pairs disruption, etc. So there's different ways you can get this full pairing, and the answer for human, what we think, is predominantly this, although there's maybe a combination of the two. So here's the cryo-EM structure that Abdallah was able to obtain, and you have the 4 domains. You have which are the mid and the peewee and the pause and the end domain, and then here's the target guide duplex. So this duplex is fairly uniform. Here's what A form would be. And then we're just morphing it to the structure we saw. So it's not perfect A form. You can see that the distance here between in the major groove at this side is quite a bit larger than A form, and then this gets narrower here on this side. So we see some distortion of the duplex, but still, not a huge bend or anything like that either. So what's also happening here is a movement of the end domain. So this moves about 10 to 12 angstroms. And so I'm just going to show you that morph again. And this time, last time we were focusing on the RNA. You can do that again if you want to, but at the end, we're going to see a shift of this end domain. So here it goes. You get this twisting, everything's even but then at the end, you can see the end domain shift up and out, and also a movement of the pos domain. So about 10 to 12 angstroms. So it's a combination of the change in the distortion in the RNA and the end domain shifting up out of the way. And as the end domain shifts, and then of course you're making the active site. So that shifted end domain exposes some basic residues that we thought might be perhaps interacting with the duplex. We mutated them, and mutating them individually we didn't really see much. But then if we mutate all of them, we can start to see maybe a six-fold difference. So they're not having a huge effect, and we don't actually know which of them are having a beneficial effect. If we mutate a couple of them to acidic residues, you also see about a 10-fold difference. So I would say a modest potential effect of these residues. And Ian's going to talk tomorrow, and based on his subsequent work, if it's probably these two that might be mediating this effect, but we can't say from our work. And then just thinking about the conformational changes in the protein, just one more morph here. The central gate here, you can see it's moving apart, and as it does, it makes room for the RNA here. So here, the set of contacts. Many of these have been seen previously. Like, again, the guide here is in red, the target in blue. These seed-based contacts have been seen previously for the seed pairing structures, and then these contacts here to the middle region were pretty much analogous to the ones that were seen in plants, but it's nice to also have a look in the human. And then here are these potential interactions involving side chains from the end domain. In our structure, we just don't have the resolution to be able to actually model the side chains, so it's kind of fuzzy here for us. We also, looked at the residues that were interacting near this purine pyrimidine. There were 3 of them, and so we mutated them and see if we could lose this preference for the purine in the guide RNA. And what we found was that, in the wild type, you have this a little over threefold preference for the AU versus the UA. If we mutate other nearby residues, here 712 or 635, you still see that preference, but when we mutate 710, we no longer get that preference. So it looks like a contact from 710 is helping to mediate this preference for purine pyrimidine at this position. Okay, and as I mentioned before, tomorrow morning you'll get the treat of hearing about Ian's structure, which they have published in this preprint, and there you can see that there's lots of similarities. One difference is that in their case, the pos domain basically looks like it's structurally uncoupled. It's so flexible, it's structurally uncoupled from the rest of the protein that it's not part of their structure. And we had to go to quite an extent, to see this. These two domains here are very flexible, and you can see, you can see that in the cryo-EM data. But another important difference is that look at the distortion here in the duplex. You can see that there's quite a bit more. This is quite a bit wider, and this is a bit narrower than what we see here. So they see a larger contribution for helix distortion than end domain repositioning. So it'll be really interesting, once their coordinates are available to be able to compare these structures and then consider whether the differences might be due to just different guide RNAs or different protein sequences or different conditions or whatever. Okay, so just to summarize then, we find these three sequence determinants that help explain the differences in slicing rate, that help explain why you can get 150 or 250-fold difference in slicing rates depending upon the guide RNA sequence. We see that this two-helix intermediate appears to be important for achieving the paired configuration. That this relationship between the importance of having 3' pairing and the stability of the pairing here in the middle implies this conformational change is important to get of this helix rolling to get to this configuration. And this fully paired configuration is not intrinsically unstable. Okay, you can see it by chemical probing for a lot of different sequences, and so that end domain doesn't have to act as a wedge to disrupt the terminal pairing. And in fact, the end domain can move out of the way, and then that plus distortion of the helix can accommodate the fully paired target for the human ego. And then also there's this central loop where the 710 side chain or amino acid comes from, and that helps to mediate the slicing rate and as well as this possession 10 preference. Okay, so with that, I just want to thank the people who did the work, Peter Abdallah, who was in Seychelles lab, and thank you all for your attention.
Thank you, David. David, really nice talk. I really like the thinking. How mucked up does it get when you put true prime mods in there and the TM goes up? Does it throw these rules out, or do these rules carry over to higher TMs?
I don't know. We haven't looked for the modified RNAs, but I'm sure people are doing that. I think I would think in principle that as you stabilize that pairing, especially in the middle, it's probably not going to. You're going to have to figure out some way of destabilizing it to get the 3' region to matter. Yeah. Again, if you're worried about slicing off targets. Yeah. So I think it is worth considering, and there might be modifications. Not all modifications would have to be stabilizing. You might choose a little bit destabilizing ones for the middle, but that's just speculation. We haven't done anything with modifications.
Hi, David. How do you think that this would translate over to microRNAs where you don't have it fully paired? Do you see more of the successful microRNA binding sites having that 3' region right where you see it in these things?
So the seed pairing plus supplemental pairing was actually inspired by what had been observed by with microRNAs. And many people think of microRNAs always having that supplemental 3' pairing. I would argue that only about 5% of the time that you could see a conserved additional pairing at the 3', in the 3' region, that supplemental region, whereas almost all the time you have the conserved seed pairing. So with the two-helix structure, that's very microRNA-like, and I think everything there would certainly apply to microRNAs. I'm not sure I'm answering your question though. Yeah, sorry. Hi.
Can you hear me now?
Yes.
Okay. So I have a question about endogenous mRNA regulation. And so is there a correlation between codon usage and if an mRNA is more or less regulated based on this finding?
We don't. Most of the interactions between microRNAs, the biological regulatory interactions between microRNAs and mRNAs, are in the 3' UTR, and so that's where most of the binding action is. There is some activity for binding in the coding region, but it's about overall. It's about 6 times less effective because the ribosome is bumping off the silencing complex. And so we don't really study the coding as much because it's so ineffective, so I can't really answer the question about codon biases. Yeah.
I'm really struck by this huge range for K-slice that Peter observed. But then when you do the similar experiments in the cells, it seems like the range is compressed. Is that because you couldn't find the right sequence? The sequences are different, or is there something else going on?
Yeah. So in cells, I forget. Maybe it's a twofold difference. I don't know if I'm doing justice to it. Maybe let's say it's a twofold difference. I think that or even if it's 3 or 4, it's not 250-fold. So I think that once you're fast, it may not help to be that much faster. And so those differences that we're seeing, I think, were probably between very slow and medium. And so you're only capturing part of the dynamic range. I think that's part of it, but I have to think about it more if might be something else. Yeah.
So there is some ongoing discussion for many years now about cooperativity of different 3' complex binding to 3' UTR. So if we believe this is true, what parts of the cleavage or binding or release being affected?
Yeah, we've looked at that cooperativity with respect to the seed pairing type of recognition, where you're having the dedylation, decapping decay, and that what we see is that if the 2 sites are not on top of each other, at least 8 nucleotides from each other, so you don't have interference, and then out to about 40 nucleotides. If they're between 8 and 40 nucleotides from each other, you do see a pretty strong cooperative effect of this seed-based type targeting, and we think that that's mediated by this next protein, the TNRC6, the adapter protein, that can make interactions to both of those silencing complexes. And if they're near to each other, it'll be interacting with both of them, and so they're each helping each other stay bound. With respect to slicing, I think that's probably what you're most interested in. I would think that TNRC6 could help the association rate of a risk complex, but I think in most cases that probably isn't limiting. And so I don't know how much, and the slicing is usually pretty fast, and there's no real off rate, as far as I know, for a slicing site. So the chances for cooperativity to help you seem quite diminished. But I can't think of experiments that have addressed that. Maybe you're thinking of some, but right now I can't think of any. Yeah. Do you?
Why? Tells who. As long as Mana gives me a microphone, I can ask another question. So if you look at in vivo data from Alnylam and others, very often there's a weird phenomena which called dose-dependent plateau. And even if the doses where you can get like 95% silencing, but at certain doses it's not there, but it stays for extended periods of time. And we know that RIS complex is saturated. What do you think is, from a kinetic perspective, is defining this plateau phenomenon?
Is there any chance that the RNA is still in the nucleus? Or has that been ruled out?
No, it's not that. It's true for even mRNAs, which are fully cytoplasmic.
Yeah, maybe Kevin can answer that question. You can ask him that one.
He's going to answer.
Since you already asked that, that was my second.
David, I have a quick question following Steve's point. At the end, when you go instead of AU base pairs, if you make them all GC, that itself can make a big difference.
Yeah.
Has that been quantitated in your studies, and how that kind of base pairing affects?
Are you talking about just in the central region?
No, in the 3' region.
Oh, where you have
Strong GCs everywhere.
Yeah, I think we haven't looked at that. I think that one issue you're going to have there is product release.
Yes.
And so that could be a concern.
Right.
Yeah.
Similarly, the G quadruplexes also can play a role in the secondary structures.
Yeah. Yeah, no, there's all kinds of things happening with the RNA structure that is going to affect the association rate, the K-on. Yeah, for sure.
Oh, please.
Not yet. I can hear you, but
Thank you. Okay, perfect. Thank
Good.
you. So in the case of those microRNAs where there is pairing in the supplementary region, one can imagine that in those central regions there might be kind of non-canonical or non-Watson-Crick pairing that might sort of induce this role and then kind of lead to this sort of secondary structure that you see. Is that something that you imagine would be possible and also like potentially different in terms of the mechanism of a microRNA where it has that supplemental pairing or not?
well, I think each microRNA has a few of its targets that have that supplemental pairing. And whether, in the central region that's not as paired, whether there are non-canonical interactions there could be in principle, although it seems like the guide RNA really isn't available for pairing because it's, and if you were pairing, you'd have to wrap between the guide RNA and the protein, which is going to be tough. And so it's really that seed pairing and supplemental pairing that are the really easily accessible interactions to the guide. And I think what's happening in the middle probably not that much opportunity for non-canonical pairing, and it's just when you get that second helix rolling that you can now get pairing into the middle. But that you can't really do that with non-canonical interactions. I think they're not going to be as energetically favorable, but also if you do have those, you're not going to reach the configuration where you can actually slice. I'm not sure if you're asking about that, but for you need the Watson-Crick interactions through the middle to give the conformation to get the active site for slicing. But that might be tangential to what you're asking. Yeah.
Okay.
We have, speaking of Alnylam, we have a comment, nice work from Yakanta, and she's wondering how the slicing rate of 6S was monitored.
How the slicing what?
Of 6S.
Oh, how did we monitor the slicing just for our biochemical experiments?
Yeah.
We would use radiolabeled target and then just run gels looking at the remaining target and the product.
Last question.
Just a quick follow-up. I was wondering, I guess it's assumed that the release of the cleavage product is not usually kinetic bottleneck here, but is that always true, and is that also depends on the pairing strength?
Yeah, I think that this product release can definitely be rate limiting. It's really easy to see it in vitro where you have this burst and then very slow second phase and that's where it's just product release that's limiting the reaction. And that can also happen in cells. And you're right, it depends on the sequences. And so depending on the sequences, you can either have the association rate be limiting, the slicing rate, or the product. Yeah.
Please join me in thanking David Bartel.